Hello and welcome back to the Biochemistry for Health Sciences channel. Today's topic would be fatty acid breakdown or fatty acid oxidation. In today's video, we'll continue our discussion on early starvation, and more specifically, we'll talk about the breakdown of fatty acids. In the previous video, we talked about how glycogen in the liver is broken down into glucose by the process of glycogenolysis, glycogen breakdown, and this glucose is then exported out of the hepatocytes, and this is the glucose that maintains your blood glucose greater than 40 milligrams per deciliter, and this should be more than enough to feed our red blood cells. There are about 24 trillion red blood cells, plus to feed our brain cells. So at the beginning of early starvation, there should be sufficient glucose coming from liver glycogen to feed not only the brain cells and the red blood cells, but also most of the cells in the body. But as early starvation continues, the body makes certain that this glucose that is so vital to the red blood cells and the brain cells, this glucose would not be used up by the other cells, so that the brain and the red blood cells don't have to compete for this glucose, this very important glucose for them. So as early starvation progresses, what we now see is breakdown of triglycerides. They gradually start getting broken down. Most of your triglycerides are stored in the adipocytes, your adipose tissue. So these triglycerides are broken down into fatty acids, and of course the fatty acids can provide ATP energy to the adipocytes, but a lot of these fatty acids will be transported out into the bloodstream, and from there they should be able to provide energy for muscles, for liver, and for most other tissues. So in this way, the brain cells and the red blood cells will have this very important glucose mostly for themselves. They don't have to share it with the other tissues because these other tissues are now getting their energy from fatty acids. Also note that as we go from the postprandial state to the early starvation state, the levels of glucagon rises. This hormone glucagon will be secreted from the alpha pancreatic cells, while the level of insulin decreases. And glucagon is more of a catabolic hormone. It will stimulate the breakdown of glycogen, as well as it will stimulate the breakdown of triglycerides. So in this video, we shall talk about how cells break down fatty acids to generate ATP. So this is the part that we'll look at, how are fatty acids broken down to generate ATP. It is important to remember that there are many different types of fatty acids. Let's look at the three types that we'll focus on in this video. The short-chain fatty acids, SCFA, they have less than 6 carbons. Most of these SCFA comes from the metabolism of dietary fiber that is carried out by bacteria in our colon. We do get some SCFA in our diet, especially from plant oils as well as milk. We do not find SCFAs in chylomicrons. We do not see SCFAs in membranes, and neither do we see SCFAs in adipose tissue. So those the triglyceride in adipose tissue, the fatty acids that these triglycerides have do not include SCFA. These SCFAs, which are water soluble, can also permeate the blood-brain barrier, and they have very important functions, not only just in providing energy, so they act as fuel molecules. They also are involved in a lot of signaling functions as well as regulation of metabolism. Here is an example of SCFA, short-chain fatty acid. You can see we have the carboxyl group and a total of 4 carbons. So this is butanoic acid or butyric acid. Then we have the medium-chain fatty acids, MCFAs. There are about 7 to 12 carbons in the MCFA. They come from our diet. We find them in various plant oils as well as in milk. They are quite water soluble, so they are not found in chylomicrons. There is a small amount of MCFA that we see in human adipose tissue. Like the SCFA, they can also permeate through the blood-brain barrier, and they have signaling functions, regulation of metabolism, and they also serve as fuel. In the clinical world, we do actually use triglycerides which are rich in MCFA. We do use these emulsions for feeding patients that have impaired digestion. As we discuss the breakdown of fatty acids, you'll see why MCFA is better in this respect, feeding patients with impaired digestion, compared to, let's say, the use of LCFAs. So here's an example of a medium-chain fatty acid, MCFA. This one, once again, you have a carboxyl group. And in this case, you have 8 carbons, so this is octanoic acid. The common name is caprylic acid or caprylate. And then we have the LCFAs, long-chain fatty acids. These are the most abundant, the major fatty acid, and that's what you usually see in your textbooks. So these have about 13 to 21 carbons. They're found in diet, and as mentioned, they're the major fatty acids of triacylglycerol that is found in adipose tissue. So most of the fatty acid in the triglycerides of adipose tissue is LCFA, a little bit of MCFA. Unlike the SCFA and the MCFA, long-chain fatty acids cannot permeate through the blood-brain barrier. And shown here is a very common LCFA that has 16 carbons. So this is palmitic acid, or in the anion form, that would be palmitate. There are several other fatty acids, such as your unsaturated fatty acids, whether they are monounsaturated, polyunsaturated, or fatty acids that have an odd number of carbon atoms. And these type of fatty acids, we'll talk about the oxidation or the metabolism of these fatty acids in the next video. So let's look at the key steps in the breakdown of fatty acids, what we call as beta oxidation. So the main focus in this video is to talk about early starvation. So we are going to look at this part here, the breakdown of triglycerides into fatty acids in adipose tissue, the release of fatty acids into the bloodstream, and how these fatty acids are then used to generate ATP. Remember, the major fatty acid that you find in your adipose tissue is almost entirely LCFA, and there's a little bit of MCFA. We shall also talk a little bit about the SCFA as well as the MCFAs that come from the diet. So we will discuss a little bit about that, because this is a good place to compare their metabolism, their breakdown, compared to the breakdown of LCFA. So we begin this story of the breakdown of lipids that will give us ATP in early starvation, that starts in early starvation, and we'll begin this story right here. The breakdown of triglycerides into fatty acids in the adipocytes. So the adipocytes have several types of lipases. We have the adipose triglyceride lipase, ATGL, that breaks down triglycerides or triacylglycerols into diacylglycerol and free fatty acid. Then we have hormone-sensitive lipase, HSL, that will break down the diacylglycerides, diacylglycerol, into monoacylglycerol and free fatty acids. And then we have the monoglyceride lipase that will break down the monoacylglycerol or monoglycerides into glycerol and free fatty acids. So at the end, we will have the free fatty acids. Remember, most of these free fatty acids would be LCFA. A little bit would be MCFA, but most LCFA. And we'll also have the glycerol produced in the adipocytes. And this process starts when your glucagon levels start rising as you go from the postprandial state into the early starvation state. Now, the LCFAs accumulate in the adipocytes, and they are exported out of the adipocytes either by passive diffusion as well as by proteins such as the fatty acid binding proteins. These particular FABPs you find in the plasma membrane, as well as proteins such as CD36. The small amount of MCFA that are formed from the breakdown of the triglycerides in your adipocytes, these MCFAs probably get out of the adipocytes, out of the plasma membrane of adipocytes, by passive diffusion. Now, glycerol is very water soluble, so there's no way it's going to get out of this membrane of the adipocyte. So it will require a transport protein, and the transport protein responsible for exporting glycerol out of the adipocyte is aquaporin 7 or AQP7. Now, it is believed that this glycerol that is formed in the adipocyte, it doesn't really accumulate in the adipocyte because of two reasons. One is adipocytes have a very low activity of glycerol kinase. This is the enzyme that makes glycerol into glycerol 3-phosphate, thereby trapping the glycerol inside the cells. And the second reason is this glycerol is very efficiently exported by this AQP7. This is a very efficient transport protein that transports glycerol out of the cell. So this means the glycerol that is formed from this breakdown of triglycerides is really not reused by the adipocytes. Instead, it is exported out of the adipocyte. So once the glycerol comes out of the adipocyte, it will go into the bloodstream. Now, glycerol being water soluble, it simply dissolves in the plasma of your blood. So once it's in the blood, it will go to the various parts of the body, and your body cells will take up this glycerol into the cells by proteins such as aquaporin 9, AQP9. Now remember that during starvation we are not eating, so the glucose during starvation we talked about comes from your liver glycogen, but then later on we'll talk about this in a future video. The body actually starts making new glucose, and this process is called gluconeogenesis and happens mostly in the liver and a little bit in the kidneys. And we'll see this process of gluconeogenesis in a future video, but glycerol, for now, just understand that glycerol is a very good chemical for making glucose because it is neutral, doesn't have any charge, and it doesn't have any nitrogen. So it's a very nice chemical for making new glucose, which will be very important during starvation to feed the red blood cells as well as the brain cells. So we'll talk about gluconeogenesis during late starvation, because that's where it becomes very predominant, although it starts in early starvation, but we will talk about it in late starvation, and we'll see how the glycerol is converted to the glycolytic intermediate dihydroxyacetone phosphate by these two reactions here, glycerol kinase, which makes glycerol into glycerol 3-phosphate, and then glycerol 3-phosphate dehydrogenase, which will take the glycerol 3-phosphate and make it into dihydroxyacetone phosphate. And then this dihydroxyacetone phosphate is made into pyruvate by glycolysis, and this pyruvate is made into new glucose by the process of gluconeogenesis. And as mentioned, this happens mostly in the liver, a little bit in the kidneys, and this new glucose that is made in the liver and kidneys will then be sent out into the bloodstream to feed the brain and red blood cells that depend so heavily on glucose. Okay, so we saw what happens to the glycerol that is produced from the triglycerides in your adipocytes. Now let's see what happens to the fatty acids. Okay, so now the long chain and medium chain fatty acids, once they are in the bloodstream, they will be bound to albumin because their solubility is not as high as the short chain fatty acids. So the short chain fatty acids are very water soluble. They don't need any binding to albumin. However, the LCFAs and MCFAs will be mostly bound to albumin. So the concentration of fatty acids in your bloodstream will gradually be increased. It will gradually go up as we go from the postprandial state into starvation. So about 12 to 24 hours after a meal, you will see your fatty acids in your bloodstream increasing to significant levels. Now, how do these fatty acids enter cells in the various parts of the body? For SCFA and MCFA, we do not need any transport proteins. They simply will go through the plasma membrane by passive diffusion. However, for LCFAs, for long-chain fatty acids, we have identified some transport proteins that are important in taking up LCFAs. These transport proteins include fatty acid transport proteins, or FATP. We'll talk about this part in a few seconds. It's interesting that these transport proteins not only transport the long-chain fatty acids, but they can also convert the long-chain fatty acids into their acyl-CoA form. We call this the acyl-CoA synthetase activity, which we'll talk about in a few seconds. And then we have the plasma membrane FA binding protein, or FABP PM. PM here stands for plasma membrane. Another protein that has been identified that plays an important role in the uptake of LCFA by cells is the fatty acid translocase, FAT or CD36. Once the LCFA is in the cytoplasm, it will bind cytoplasmic fatty acid binding proteins to be shuttled around in the cells. Once again, this is because LCFAs are not water soluble. On the other hand, SCFAs and MCFAs, they do not need any binding proteins in the cytoplasm because they are quite water soluble and they should be able to travel around inside the cells themselves. So now that the fatty acid is inside the cell, in this case now in the cytoplasm, now we are ready to start breaking it down. So the first reaction here is the activation of the fatty acid. Basically, this carboxyl group is converted into a thioester, so it joins a coenzyme A to form fatty acyl-CoA. So a CoA is required, ATP is required, and the way this ATP is broken down, remember most of the times when we talk about ATP usage, how is ATP broken down? It breaks down into ADP and PI. And we say we used up minus 1 ATP. But in this case, we are breaking ATP into AMP and PPI. So this is a different way of breaking ATP, and in this case it's equivalent to using minus 2. So we're really using 2 ATPs to activate this carboxyl group into a thioester. The enzyme that catalyzes this reaction is called acyl-CoA synthetase, ACS. And for LCFAs and MCFAs, we use acyl-CoA synthetase that are present in the endoplasmic reticulum or the outer mitochondrial membrane. For the SCFA and MCFA, that is, which are less than 8 carbons, so the MCFAs that are less than 8 carbons, they can enter the mitochondria, and in the matrix they will be converted or activated by enzymes, the same enzyme acyl-CoA synthetase that is found in the matrix. So we have these enzymes found in the matrix, we have them in the outer mitochondrial membrane, and we have them in the ER. So the next step will now be the transport of this fatty acyl-CoA, these activated fatty acids. We have to transport them from the cytoplasm into the mitochondria. Remember, if they are 1 to 8 carbons, that is not necessary because they can just get into the mitochondria by themselves. So for fatty acids 1 to 8 carbons, that's not necessary. But if the fatty acid has more than 8 carbons, then we need a special way to bring these fatty acids from the cytoplasm into the mitochondria. So the system we use, the method we use, the cells use to transport fatty acids that are more than 8 carbon from the cytoplasm into the mitochondria, this transport system is called the carnitine transport system. And this carnitine transport system has 3 parts. So there is an enzyme, carnitine palmitoyl transferase 1, CPT1. Then there's an actual transport protein called carnitine acyl-carnitine translocase, CACT. And then there's another enzyme, carnitine palmitoyltransferase 2 or CPT2. So let's look more carefully at the transport of these fatty acids with more than 8 carbons from the cytoplasm into the mitochondria using this carnitine transport system. First of all, what is this chemical called carnitine? Now, carnitine, you see the structure here, has a quaternary ammonium. This is a quaternary ammonium compound, and you can see these compounds are positively charged. Most of the carnitine comes from the diet, although we humans can also make carnitine. You can see from the structure, this is a relatively small compound, has a positive charge here, has a hydroxyl group here, has a carboxyl, there's negative charge, very, very water soluble. So there's no way this compound is going to enter the plasma membrane by itself. It will need the help of a transport protein, and that transport protein is OCTN2, organic cation transporter 2, or OCTN2. As we shall see in a few seconds, this hydroxyl group is the one where the fatty acids will be attached or detached to carnitine. In the first step of this transport of carnitine from the cytoplasm to the matrix, we have the fatty acyl-CoA reacting with carnitine to form fatty acyl-carnitine and releasing coenzyme A, and this reaction is catalyzed by the enzyme carnitine palmitoyltransferase 1 or CPT1. In the next step, the fatty acyl-carnitine is transported into the mitochondria by this inner mitochondrial membrane protein called carnitine acyl-carnitine translocase or CACT. So that's how the fatty acyl-carnitine comes into the matrix through this CACT. In the last step of this transport, the fatty acyl-carnitine will react with the coenzyme A, and that will give us the fatty acyl-CoA. So you can see the fatty acyl-CoA from the cytoplasm is now fatty acyl-CoA in a mitochondrial matrix. The carnitine is released, and this carnitine is transported out by the same transport system, the carnitine transport system. This reaction here is catalyzed by carnitine palmitoyltransferase 2 or CPT2. So this is how we bring in fatty acids that have 9 carbons or more than 9 carbons from the cytoplasm into the matrix. Quite a long process, and that's why fatty acids that have 8 or less carbons, they don't need to have this transport system. They can go directly into the mitochondria, and that is one reason why we can get energy from these fatty acids much more readily than we can from the longer chain fatty acids. Now, the reverse transport is an important thing to note. The fatty acyl carnitines that are formed in the matrix can be sent out when they accumulate. They can be sent out by the same transport system into the cytoplasm, and then from the cytoplasm they are exported out by some protein that we haven't identified yet, and they come out of the cell, and then they can actually enter the bloodstream. So in the clinical world, we do actually have a test called the plasma acyl carnitine test that is used to diagnose or monitor various metabolic disorders of fatty acid oxidation. Now that the fatty acyl-CoA is in the mitochondrial matrix, we are ready to start the oxidation or the breakdown of these fatty acids. So in the first reaction that happens in the mitochondria, the enzyme acyl-CoA dehydrogenase, we'll just abbreviate it as AD, this enzyme will form a trans, and that's important to know, trans, not a cis, because remember most of the double bonds we have in fatty acids are cis, but this one here is trans. And this becomes important in the next video when we talk about the breakdown of unsaturated fatty acids. So this is a trans double bond that is formed between carbons 2 and 3. In other words, between the alpha carbon and the beta carbon. This enzyme uses FAD. So the FAD is reduced to FADH2, and the electrons from this enzyme, from this FADH2 that is bound to the enzyme, is transferred to ETF. ETF is a protein, electron transfer flavor protein, and then these electrons then go to ETF dehydrogenase, ETFDH, and this enzyme will send the electrons to coenzyme Q. Remember, coenzyme Q is one of the mobile electron carriers of the electron transport chain. Okay, so basically that's how the electrons are carried from this acyl dehydrogenase to the electron transport chain. For the long chain fatty acids, the enzyme that's responsible for this reaction seems to be this one here, VLCAD, very long chain acyl dehydrogenase, and this is found in the inner mitochondrial membrane. So this enzyme is here attached to the inner mitochondrial membrane. Now humans do have LCAD, long chain acyl dehydrogenase, but their activity seems to be very low in most cells, except for the type 2 alveolar cells, where research has shown that the activity is pretty high. Remember, these are the cells that make surfactants. But in other cell types, the LCAD activity is very low, and that's why we believe the VLCAD might be playing a more important role for the long-chain fatty acids. For the short-chain fatty acids as well as the medium-chain fatty acids, these acyl dehydrogenase, the MCAD for medium chain, and SCAD for short chain, these enzymes are found in the matrix. These are soluble enzymes found in the matrix, compared to this one here for long chain or very long chain that is attached to the inner mitochondrial membrane. So this product here that we get from this oxidation, this is a trans 2-enoyl-CoA. Trans 2-enoyl-CoA. So a double bond is between carbons 2 and 3. Trans 2-enoyl-CoA. In the next reaction, water is added to this trans 2-enoyl-CoA to give you this product here, 3-hydroxyl acyl-CoA. You can see the water is basically added across the double bond, and the OH part of water goes to carbon number 3 here, and carbon number 2 takes the hydrogen part of the water. This hydration reaction, the adding of water across the double bond, is carried out by the enzyme called enoyl-CoA hydratase, which belongs to the lyase family. In case of long-chain fatty acids, this enzyme, enoyl-CoA hydratase, is actually part of a bigger protein called mitochondrial trifunctional protein, or MTP. and this MTP is attached to the inner mitochondrial membrane. However, for SCFAs and MCFAs, this hydratase is soluble and is found in the matrix, and this hydratase is called crotonase. So crotonase is for SCFAs and medium chain fatty acids. This enzyme is found in the matrix, while for long chain fatty acids, the hydratase enzyme is part of MTP, and this enzyme is attached to the inner mitochondrial membrane. In the next reaction, the hydroxyl group is oxidized to a carbonyl, and in this case the coenzyme that is used is NAD. So carbon 3 now is a carbonyl, and this product here is called 3-keto. Carbon 3 is keto, so 3-keto acyl-CoA. The enzyme that catalyzes this reaction is 3-hydroxyl acyl-CoA dehydrogenase, and this enzyme for long-chain fatty acids is again part of the mitochondrial trifunctional protein, or MTP, that is attached to the inner mitochondrial membrane. However, in the case of the short chain and medium chain fatty acids, this enzyme, this dehydrogenase enzyme, what we call short chain 3-hydroxyl acyl-CoA dehydrogenase, or SCHAD, this is a soluble enzyme found in the matrix. So if we consider these reactions from 6 to 8, we see this carbon number 3, this C3 here of the fatty acyl-CoA, is converted from a CH2 to a CO. So this carbon 3, remember, is also called the beta carbon. This is your alpha carbon next to the carbonyl. So beta carbon becomes from CH2, is converted to, is totally oxidized to CO. That's why we call this fatty acid breakdown as beta oxidation, because the beta carbon is being oxidized from a CH2 to a CO. The NADH that is formed in this reaction will again donate its electrons to complex number 1 of the electron transport chain. In the next step of this beta oxidation, this keto-acyl, this 3-keto-acyl-CoA, is broken. The carbon bond between number 2 and number 3 is broken. So we get this part here, RCO, and since we are breaking this with coenzyme A, this part becomes RCO attached to coenzyme A. This is your fatty acyl coenzyme A. And then this part here will become, this one here that has 2 carbons, 1 carbon, 2 carbons, this will become acetyl-CoA. So we are breaking down the keto-acyl-CoA into a fatty acyl-CoA which has 2 carbons less, and an acetyl-CoA. The enzyme that catalyzes this reaction is called 3-keto-acyl thiolase. In case of LCFA, this thiolase enzyme once again is part of the MTP. However, for SCFA and MCFA, the enzyme is called medium chain keto-acyl-CoA thiolase, or MCKAT. And this MCKAT, once again, is a soluble enzyme found in the matrix. So this is how a fatty acid is broken down. So between carbons 2 and 3, this part comes out as acetyl-CoA, and then the remaining part here comes out as the fatty acyl-CoA that is reduced by 2 carbons. So these 4 reactions, 6, 7, 8, 9, this form what we call a beta oxidation cycle. So that's one cycle of beta oxidation. The fatty acyl-CoA that is formed now, that is shortened by 2 carbons, this will now undergo back to reaction number 6 and start the second cycle. So there'll be another cycle, and that cycle will break down once again, take away 2 carbons, and then the product that is formed once again will be shortened by 2 carbons, and that product will go through another cycle, and this continues until all the fatty acid is broken down into acetyl-CoA. So an easy way to remember these 4 very important reactions, oxidation, hydration, oxidation, thiolysis, is OHOT, O-H-O-T. So let's summarize the beta oxidation. Let's use an example of palmitic acid. Now remember, palmitic acid has 16 carbons, therefore it's a long chain fatty acid. So if this palmitic acid was to go through the beta oxidation, of course the palmitic acid will enter the cytoplasm, and there will be converted to palmitoyl-CoA. Okay, so that's what we have here, palmitoyl-CoA. Now, being a long chain fatty acid, it will be acted on by the enzymes that act on the long chain fatty acids, and that would be VLCAD and MTP. And let's say 3 cycles later, so from a 16 carbon, 3 cycles means you're removing 6. Remember, every cycle remove 2 carbons, so 3 cycles means you're removing 6 carbons. So from a 16 carbon long chain fatty acid, after 3 cycles, it will become a 10 carbon medium chain fatty acid, decanoic acid. So now that it's a medium chain fatty acid, it will be acted on by all the enzymes that work on medium chain fatty acids, your MCAT, crotonase, SCHAD, and MCKAT. And from here we have a 10 carbon fatty acid, so 2 cycles later, so that's -4, that will give you a 6 carbon fatty acid. Now a 6 carbon fatty acid falls into the SCFA region, so this is now a short chain fatty acid. Okay, so hexanoic acid, 6 carbon fatty acids, also called caproic acid. So this caproic acid, being a short chain fatty acid, will be acted on by the enzymes that work on short chain fatty acids, SCAD, crotonase, SCHAD, and MCKAT. Okay, so 6 carbons. After 2 cycles, you would get 3 of this acetyl-CoA. And that's how a palmitic acid, which is a long chain fatty acid, will be acted on first by the long chain enzymes, and then by medium chain enzymes, and then by short chain enzymes, eventually breaking down the palmitic acid into acetyl-CoA. In an earlier video, we calculated how many ATPs we get from glucose, because ATP is energy, so it's very important to understand how much energy we get from a certain chemical. Here, let's do the same thing. We'll calculate how many ATPs we get from a fatty acid. And the most common fatty acid is palmitic acid, 16 carbons. So let's calculate how many ATPs we get from a palmitic acid. One way of doing this is calculating the number of cycles. Okay, and that can be done by number of carbons minus 2 divided by 2. So palmitic acid has 16 carbons, so 16 minus 2 divided by 2. So that's 14 divided by 2, and that gives us 7. That means to break down palmitic acid into acetyl-CoA, we need 7 cycles. Now each cycle of beta oxidation produces 1 FADH2 and 1 NADH. Therefore, 7 cycles will produce 7 FADH2 and 7 NADH. Now remember from a previous video on the electron transport chain oxidative phosphorylation, each FADH2 gives you 1.5 ATPs when it goes to the ETC-OP. NADH gives you 2.5, so 7 times 1.5 gives you 10.5, 7 times 2.5 gives you 17.5 ATPs. Now remember, we get ATPs not only from FADH2 and NADH, but we also will get ATPs from the acetyl-CoA. So how many acetyl-CoAs are produced from 16 carbon fatty acids? And the answer is number of cycles plus 1. Since there are 7 cycles, add 1 to that, that gives you 8 acetyl-CoAs. And that makes sense because palmitic acid is 16 carbons, acetyl-CoA is 2 carbons, so you can see 16 divided by 2 is 8. So we get 8 acetyl-CoAs. Each acetyl-CoA will go into the Krebs cycle and then produce NADH, FADH2 as well as GTP, and that will give you 10 ATPs. So each acetyl-CoA gives you 10 ATPs, so 8 will give you 80 ATPs. So we add all this up, 10.5, 17.5, 80, add all this up, we get 108 ATPs. To activate the carboxyl group, remember the activation of the fatty acid, we use ATP, but this ATP was broken in a way that gives you AMP and PPI. So this means we use an equivalent of 2 ATPs, not 1 here, 2. So we must subtract 2 from 108, and that will give you 106 ATPs. So there is a net of 106 ATPs that are formed from 1 palmitic acid. So you can see that every 2 carbons, if this is a figure here with your palmityl-CoA, so count 1 carbon, 2 carbons, we break this here, 1 carbon, 2 carbons, we break this here, 1 carbon, 2 carbons, break, 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 and then you have 4 carbons left. The last cycle, when you break, you will get these 2 fragments here. And you can see if you count all the cycles, you use a total of 7 cycles. Now we can calculate the amount of ATP we get from a C6 fatty acid. Why C6? Because glucose has 6 carbons too. So let's compare this carbohydrate glucose, the most important fuel, with 6 carbons, with a fatty acid with 6 carbons, caproic acid or hexanoic acid. So if you go through the same process as what we did before, you will see that you get a net of 36 ATPs. Okay, compare that with glucose where you get about 30 to 32. So clearly fatty acids give you more ATP compared to carbohydrate like glucose. That's because the average carbon in a fatty acid is CH2, which is more reduced, while the average carbon in a glucose was CHOH. You can see this is partially oxidized. So the carbon in a carbohydrate is partially oxidized. Carbon in a fatty acid is more reduced. That's why you get more energy. But remember, to the red blood cells and to the brain, it does not matter that the fatty acid is a better source of energy than glucose. because red blood cells just do not have the machinery to break down fatty acids. And to the brain, it can get some energy from fatty acids, but it's believed that fatty acids, the intermediates of fatty acids, as well as fatty acids themselves, can be quite toxic to cells. And that's why the brain prefers not to use fatty acids. So the brain will get about 20% of energy from fatty acids, but most of the energy, 80%, will come from glucose. So there we have it. Now we have talked about providing glucose to the bloodstream in starvation. That's coming from your liver glycogen, and this is great for your red blood cells and brain cells. And then we now finished talking about how we can take fatty acids that are stored as triglycerides in the adipocytes and how tissues can get ATP from fatty acid breakdown. So we can break down fatty acids to give us ATP. So that's great. That means while the rest of the body is using fatty acids, this glucose here can be reserved for the red blood cells and the brain. So this continues to happen in early starvation, and as the starvation continues and continues, if you do not eat when you get up in the morning, your breakfast, you will gradually transition into late starvation that we'll talk about in future videos. In the next video, we shall look at the oxidation or the breakdown of other types of fatty acids. So until then, once again, thank you very much for watching this video and supporting this channel, and have a very happy and healthy day.